getting towards the end of the course if we're talking about publishing images. So the first thing that we need to do, and that is really important, is to read the journal guideline. This is because each journal will have slightly different guideline. I think the journals have been better over the years at putting some guidelines, and guidelines that make sense is not always the case. I think they're a little bit stuck between the printed world and the digital world very often. So there are more and more journals that are only digital. And that is sort of creating some confusion a little bit. So I recommend really reading the guideline of the journal. And then also there are a few links that I put here, some online tools to help you deal with your images and how to make figures, and also some a very nice blog posts from Journal of Cell Biology that can help you with some advice. So first, let's talk about the different aspects, let's say, of publishing images. The intensity, to start with, we need to have no bleed-through, no saturation, no underexposure in the area of interest, and we need to fill in the histogram as much as possible. So what does that mean to fill in the histogram? It just means that you should be just below the saturation point and just above the underexposure. But as you know, it's most of the time our histogram looks like this, means most of the pixels are on the dim side of the image. But that's totally okay, because we have noise and all sorts of stuff that fills up the whole thing. So if we look at this image, to start with, I want to tell you that there is something a bit funny in Fiji. Fiji considers the size of the image. And so at the top of the image here, it will tell you it's a 16-bit image, because as far as the computer is concerned, it's only 8 or 16. So everything that is between 8 and 16 is considered as, for the computer, the size, the file of the size, as 16 bits. So Fiji will tell you at the top here that it's a 16-bit image. But if you look at the image info, you will see that it was acquired, in this case, in 12 bits. So if you have 16-bit here, it doesn't mean it's a 16-bit image, actually. You need to look at the image info. So then you see this by clicking on Control-I in Fiji. So this is the information in the image. We see now it's a 12-bit image, so it means from 0 to 4095. And so when we display all the data, it's like that. So it looks quite dim. And it is from 0 to 4095. We display everything. Okay. Now, if you click on Control-H, you will get to the histogram, which is here, where you can see the minimum and the maximum intensity in the image. So this is only what is possible, but this is the minimum and maximum in the image. When we see something like this, when we are like above half of the maximum, that is quite good. You want to be, it means that you're above 0, means you have no underexposure, and you're you're not at 4095, means you have no saturation. So this is very good. And really, in general, you would want the brightest pixel to be around 2/3 of the full range. Or you can have more, but if you have a Z-stack or a time-lapse or something, you want to be quite conservative. So a good maximum would be around here. And clearly, if you only acquire one image, you can make it in such a way that you are just below saturation. That's also okay. But for Z-stack, be more careful. So this is what it means to fill the histogram. It means like where the maximum is as close as possible to the maximum of the bit depth. I remind you that if you want to compare intensity between images, they must be acquired in the same with the same settings exactly, including the bit depth. And in that case, the acquisition, you must set the settings for the brightest sample, so that you do not have saturation. So all the settings set for the brightest, and then you image all the others. So now the orientation of the sample is also quite important in when you want to compare images and want to display images in an article. So unfortunately, if you rotate a digital image, you're going to do something that is called extrapolation. And it means that there will be some distortion if the image, except if the rotation is 180 or 90 degrees or 370. So if it's a multiple of 90 degrees, you have no distortion, but if anything in between, you have some distortion. So how to deal with this? There are some ways where you can, so if you have like the type of sample where you must orientate, for example, if you have a long and slim sample, like a piece of skin or zebrafish, something like this, you would want all your images to have the same orientation in your article. The best way is to rotate during acquisition, before you acquire your image. And this is something that is very, very simple to make with these custom-made 3D printed stage insert that rotate. Before you acquire your image, you can turn the sample in such a way that you acquire already with the correct orientation. This is the very best way, because then you don't have to deal with all sorts of stuff afterwards. And certainly you do not have to rotate the image afterwards, which may create distortion, especially if you have small objects, then it will be visible. With big objects, it doesn't really matter. So, and if you need this, we know a guy who does it, so you can ask me, and then I will put you in contact with him. So the acquisition settings now, when you want to publish. So we need to use different settings for different purposes. We have seen so far that if we want to acquire an overview of the sample, we're going to try to go as fast as possible and everything. Then we want to acquire images for data. We will probably use different acquisition settings. Well, if you want to acquire an image to publish it in the paper, it's likely that you will need, again, different acquisition settings. And for what reason? If you want to go for image analysis, sorry. If you want to analyze the images, you will acquire images with the resolution, noise, bit depth, etc., that are needed for you to extract reliable data and answer your scientific questions. And maybe these images are very noisy, maybe they're very ugly, it's fine. But now, as long as they produce reliable data. Now, if you want to put such an image in an article, it may not show your point, because maybe it's too noisy, you can hardly see anything. So there's no point in putting that type of images in the article. We have a different goal when we publish an image in the article. We want to show something. So then the image has to be acquired differently. So what you need to do is that the images that you acquire for your image analysis, you will load them to a data repository, so that the readers can, if they read your article and they want to access the original data, they have access to the original data there. So load your images into a data repository for the community to use as well. But the images that you want to actually publish in an article, in the figures, then they need to be, you need to ask yourself, what message do I want to convey with this image? And you acquire them to show your message. So the goal is not the same. And sometimes, so this might be the same images or not. Maybe the images that you analyze are in such a quality that you can actually publish them in an article, but very often that's not the case. And you certainly don't want to spend all your time acquiring super duper images because you think that maybe you will publish all of them. So if you acquire an image for publication, you can try to oversample, because in that case, when you magnify the image, you will not see the pixels, so the quality will be better. And this is the case, for example, if the journal, if you think you have really nice images and a cool article, maybe the journal will ask you if they can publish your paper on the front page of the journal. And in that case, you really want to have an oversampled image so that you do not see all the pixels. And the images that are smaller than 1024 by 1024 are very unlikely to make it on the journal cover because of that problem. So think about it when you want, if you think that you have a potential image for a cover, acquire with lots of pixels. And then use high illumination power to reduce the noise. You can use long exposure time to make sure everything is really bright, there's no saturation, but make sure you have a very good signal to noise ratio when you acquire an image that you want to put in a paper. And show the data image So the images that you use to acquire data, to extract data, show these images if the purpose is to show what they look like, which is sometimes the case. For example, if you use AI, you're going to want to, you have a ground truth image, and then you have which you're training your data set with. And then you have the very, very noisy images that will be then denoised by AI. Maybe you want to show these two images. That's totally okay. But if the purpose is not to show the images that you from which you have extracted your data, if you want to have a message there in your image, then you must acquire the image to show the message. Okay. So to show better images, if the purpose is to show other type of information, right? So different purpose, different settings. So the field of view, I think I said about it before, don't publish stuff like this where we have mostly black and the sample is very tiny. We can see in this image that we have these little dots here, but it's totally okay to remove them as long as, so if you crop around here and you get this data, it's totally valid as long as the interpretation of the image is not changed, okay? So this is not image manipulation to remove these two things. You're actually just cropping, and the image interpretation is not changed. This is really important. So this is irrelevant information for the interpretation of this, and if you crop, you have no change. So please do that. So you can use on the single point confocal, you can use the zoom, and on the camera-based system, you would use the crop function or higher magnification to fill in your field of view. So when it comes to the channel, we've said that, everything in black and white, but also many people image some channels systematically, even though they don't need them. Like, for example, the nucleus is typically one. Another one is the transmitted light. So you get, after a while, you forget that your transmitted light detector is on, and then you don't care at all about the settings, and you get an image like this where the transmitted light image is totally white. I say, yeah, but I don't use it. Well, if you don't use it, turn off the detector, because now you're increasing the amount of data that you have for no reason, because there's no relevant information in there. So only acquire the channels that you actually need for the analysis. Now, if we go to formats, so as you know, the image metadata contains a lot of information about the acquisition settings, like the date, the channels, the filters, the power, the laser power, and everything. This is the reason why you can go, depending on the software, but most modern software do that. You can open the image and then somehow reuse the information in the image, in the metadata, to set your microscope exactly the way it was when you acquired that image. So it's used reuse, the reload, whatever you want, but in different software. But this is a very, very useful way to go back to your settings. Unfortunately, different formats do not preserve well the metadata, so you must be very careful in preserving the metadata when you save. And also, different formats have different levels of quality and compression. So it means that many different things can be lost, like the intensity details, the resolution, and also the metadata. So always save the image in the original format that is recommended. So these are the formats that are currently in use for the Big Four. And yes, do not put your images through several changes. For example, let's say you have an image, you like it, and then you're going to be saving it as JPEG and putting it in your PowerPoint presentation, and then afterwards, oh, you don't remember where the image is. Never mind, I take it from the PowerPoint presentation, I save it again, and then I put it in TIFF and stuff. Don't. You take your original image, you make whatever you want with it for one application, and then if you want to do something else with that image, for another application, you go back to the original and you do whatever you want with it, okay? Always go back to the original. So on computer screens, we've said this before, you must have an 8-bit image, and JPEG and PNG are lossy. Well, they can be lossless as well, it depends what it is. Mostly it's lossy, so it means that there's a very high compression. and therefore the data is really scrambled, let's say. And TIFF files can be very large and slow down your presentations. So when you are presenting on a computer screen, the resolution and the bit depth is not very high, so it's quite okay to use stuff like this. When you want to publish a poster, so anything that is printed or an article, so printed or in digital form, do not use these formats at all. So you don't use that for posters and publications, and definitely you do not analyze the images or make decisions based on these formats. This is only for PowerPoint presentations on screens. For the images that are used for everything, there's one thing that is important to realize is TIFF is actually not one format. Unfortunately, TIFF is a family of formats. And so depending on the manufacturer, whatever they have set, the metadata might be scrambled anyway. So it's not easy to deal with TIFF because you never know which metadata you are dealing with. And that's a bit of a pain. TIFF can be compressed or not. It can be lossy or lossless. So it depends on the software you're using, and the metadata might be scrambled or lost partly. So do not think that because you save in TIFF, you're safe. Unfortunately, that is not the case. So only save TIFF is if this is the recommended format by the manufacturer to make sure you have the correct metadata. And for the analysis, you always use the format recommended by the manufacturer. You never do your analysis on something that you have saved in a different format. And then for posters and publications, you can use either uncompressed TIFF or TIFF with lossless compressions and 16 or 8-bit channels. These two work to make posters and publications. So when you look at the metadata, we've done that before. If you click on Control-I in Fiji, you're going to see all the metadata. And if you have just a few lines, it means you have no metadata. If you have tons and tons, you can see here the slide that would go on forever. This is good metadata that is preserved. And when it comes to the file size, you can see the compression thing. If you have here the same number of pixels and you save in TIFF or in JPEG, and here you save in 8-bit or in any other bit depth higher than 8 bits, you can see that if anything that is higher than 8 is going to have double the size as 8, but you can see how compressed the JPEG image is. It is very, very compressed. So lots of information is lost, and you will see that if you zoom into the image. So it is quite visible, the JPEG compression. So when we look again at the intensity, we've said before that we need to adjust the brightness and contrast to clearly show what needs to be seen. We have all seen articles where the figures, you can hardly see anything in the picture. What is the point, right? You have a message to convey with your figure. That's the reason why you're putting it there. So make sure that the images are very clear and that they show clearly what you want to convey. And then you try not to, you need to push the gamma if increasing the brightness and contrast looks like it's saturated. Instead, you put the gamma, so we have done that one. And all this must be mentioned in material methods. So when we change the gamma, only the bright pixels or only the dim pixels are changed, depending on the way we move it. So all the images should be in black and white with the overlay in color. And then the elements that you want to show should be very clearly emphasized with an arrow. So it should be possible for the reader to go very quickly through your figures, because this is what normally people do. They go from one figure to the next to get an overview of the article. And if you do that, you should be able to, within a second, see what you want to show in the image. And so put arrows and stuff like this so that people can understand. And be very aware of trying to make the image look good on your screen, because different screens show lots of different things. So do not push things too much on your screen, and it might look very bad when you print, for example, a poster afterwards. So when it comes to colors, we also have color blindness. It's a very common thing. So if we use green and magenta instead of green and red, then they can see. And clearly, we include also so basically most colorblind men, what they cannot use is red. They don't see red so much. So green and magenta, they will see the difference. But if you put green and red, basically they're not going to see the red. And then you can include also the channels in the black and white. And now for screen and when you want to print, there is a different what is called a color space. So we have the RGB red, green, blue color space is for screens, whereas the CMYK color space is for printing. And you can see that they are slightly different. And so what you need to do is just go to the journal requirements and see if they want a CMYK color or an RGB color. And then this is what you give them at the end. And you do the conversion only at the end. You make your figure ready, and then afterwards your image ready, and then you convert it, not before. So you do all the changes that you want to do before you convert. Okay. Now, when we publish things, we have to represent different dimensions sometimes. So there are different ways to represent a volume. One of them is the orthogonal view. And what you can do is that, so basically, this is one of the z-plane of a z-stack, okay? And here, we're seeing the yz and the xz projections. So what we're seeing is as if we had like a big book, let's say, and here you see one of the pages of the book. You can choose which one, and in here you can see the side of the book and the other side of the book. That allows you to see for this z-plane, you will see exactly what's going on inside the book. And that is a very good way to show if you want to show, for example, that an object is inside another, like a dot in the nucleus. You are going to show that in this orientation, this one and that one, it looks like it's inside, and that means it's really inside. Okay, so this is a great representation when you want to show that things are inside the other. But another way of representing a volume is to show all the tiles. Clearly, if you have 90 tiles, you're not going to do it that way. If you have just a few, you can do it this way. So it's a little bit more uncommon, but in some cases it can be useful. Another way that is used a lot to represent a volume is to use a maximum intensity projection, so MIP, it's also called MIP. So when you want to use a maximum intensity projection, what happens is that the software for each column of pixels, so again we have a Z stack there, so each column of pixels is going to find which pixel is the brightest. And then it will keep this intensity, and this in each channel. And then it will keep this intensity to build the next image, the maximum intensity projection. So for each pixel, you're going to have the intensity of the brightest pixel in the column, and then so you end up with an image that is flattened. Clearly, this only works if you have like one object, for example, here, all these red parts in here. If I have one bit here and then another one here, in the final image, they will end up at the same plane. So I will be able to see both of them. But if it's very, very crowded all over, your whole image is going to be filled with the color. So that doesn't work in that case. If you have things that are on top of each other, a maximum intensity projection doesn't work. But for many things, it works. So it's a great way to represent a volume. When we want to represent time, there is something that, so we have the possibility to put all the tiles like this of a time lapse. And we can also use a kymograph, which is what it is now. I will show you, or you can use videos, but videos cannot be inserted in the printed in the figures. Basically, they have to be in a supplementary material method, which is fine. So in this particular case, we have a cell that is dividing, and all the images have been aligned in such a way that the orientation of the cell is unchanged. And so if now we draw a line in the center here, we can measure the intensity across that line. And then when we can put this intensity in here. So you can see that we would go into dark, 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 green, 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 green and red, green and red, green and red, green, 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 dark, dark, dark. And that's exactly what you have here. So you have, now you're in the background. Here you get to the cytoplasm of the cell. Here we're in the nucleus, the other side of the cytoplasm, and then background. And once you have just these line profiles for each channel, you can align them here over time. So it means that because the images are aligned like this, it means that you are able to follow what's happening in the cell over time. And here you can see that the cell is getting out of focus because it's dividing. It normally changes plane when it does that. And then afterwards, it divides, and now we have two cells with two nuclei. So this is exactly in here. This is where we have the bridge. So, and then it happens very quickly, and then we end up with two nuclei. So this is, then we have two cells at the end, so they are completely separated. So that's a kymograph. This is called a kymograph, and it's a very nice way, if that works with your data, nice way to represent time. When it comes to video, you have to check in the you need to check in the recommendation of the journal which codec and which aspect ratio they recommend, and you have to follow this. So this is, the codec is something that you use when you create the video. So if you have a Z-Stack, you're going to save it as AVI or something like this. And for that, you have to have the correct codec. So just go to the journal and have a look at that. Data management is a big issue. We are producing more data, big data as well, and trying to figure out which one is what, where is that thing, which one did I process, how did I process it years later. This is very difficult. So when it's time to publish, maybe the questions you will have is like, yeah, I showed this very nice image in a lab meeting 6 months ago. Maybe I acquired the image 7 months ago. Where is that image? Where is the original? Yeah. Can you answer this type of questions with the way you manage your data today? So you must set up a system to manage your data quite a lot, like from the beginning, basically, of your when you start your experiment. Just make sure, if you know that your experiment is going to generate some data, image data, make sure that you have a correct way to find the data, find what you have done to it, and link it to your protocol. How did you prepare a sample and things like this. This is really important. Lots of things are not written in the metadata of the image, unfortunately. So which antibody concentration did you use? What did you use as pre-processing? And then how did you analyze the images? And then, yeah, I have the analyzed image, but where is the original? You know, all these things are really, really key and will just kill you the day you want to write an article. So data management is really important. When you use Fiji to test your pipeline, your analysis pipeline, and you try to try different things, there's one thing that is quite interesting is the macro recorder. So if you use the macro recorder, you can Google that and then you will find videos to show you how to do it. But if you use the macro recorder, every time you try something, it will write a code line. So at the end, and you can erase them if you want to go back, for example. And that means that as you are trying things, you are building your pipeline. You can save that pipeline, apply it to another image. You can save it in the same folder as the images, so to show what you have done, etc. So this is a really powerful way to be able to save what you've done in the image analysis. So you need to document all this, and you need to document any modification so that you're able to describe it later in the meta-end methods, like years later, right? So you're not going to remember. You have to rely on something else. So turn on the Fiji macro recorder to do that if you use Fiji. Many people put a lot of information in the name of the file. Unfortunately, Windows, and I don't know if it's the same for Mac, but Windows sometimes doesn't deal very well with moving a file that has a very long name. And you're going to, if it does that and it saves also the folder names, then you're in trouble. You will not be able to move your file, for example. So that can create problems. So it is much better to have all this information in the metadata of the image, if you can modify the metadata, or otherwise have a file that explains, or you can put this in your lab book. But this information must be somewhere. Putting in the name of the file is often not a good idea. This is the great advantage of Omero. In Omero, you can modify the metadata and add data there. You can add lots of different things actually to each image. So like the concentration of the antibody or whatever, you can do this on batch. So today I've imaged with that concentration, and all these images have the same. You can do this all at once. So Omero is a very nice data management tool in that sense. If you don't have that, then you must make sure you don't put it too much in the name. And then you save in the same folder the original image. So basically what is important there is that you have something systematic and that you just follow that. Set up a system. It will be much easier for you to find the original years later. So Omero is a very great tool. You can keep track of which image was used for which publication. You can share with collaborators. You can annotate. You can star the image. Starring the image is a great way for you to find it. Every day you're going to have a few images that are starred, and then this way when you want to publish it, it will be much easier for you to find them. And you can also put a link to your electronic lab book, which will help you a lot in finding how you prepared the sample and what your conclusions were on that day, etc. So we have seen an Omero figure, and it's free. There are many more options that you can do in Omero, including changing the metadata, but also the analysis. So you can have all your region of interest and everything. It's free. Illustrator is not free. But also Photoshop, the problem with Photoshop is that it is not designed for scientists. It's designed for photographers, and they don't have the same constraints as we have. So it is very easy in Photoshop to make serious errors without knowing it. Like if you're not really aware of what you're doing, and later be accused of manipulation. So you really don't want to go there, I can tell you. So when we want to publish, now if we look at the bit depths, so an RGB image is green, red, green, and blue. If we have an RGB image in 8 bits, it means we have 3 times 8 bits. So we have total 24 bits. It's because we have 8 bits per color. Now, this is a very confusing thing. When things are printed on a paper, the way it is printed, so the printing technique, it's printed as dots of ink, like this, okay, when we zoom in. And when they want to make things gray, they just simply put bigger dots, like the dancer, and then they want to go closer to white, it's just smaller dots. So we have a certain density of dots, but we have also a certain size of dots. And then we can have colors, different dots of different colors. So, and it's not possible to print anything completely black or completely white. So anyway, if your images are not saturated and not underexposed, you should not have anything that is completely black or completely white. So the thing that is confusing is that publishers tend to speak as if they were talking about printing when they are talking about the images themselves, and that can become a little bit confusing. So very often you go to the journal and they tell you, you're going to give us some images that are minimum 300 dpi. So this refers to the printing density. It's a number of dots per area unit, so dot per inch. Okay, so 1 inch is 2.54 centimeter. So number of dots per inch, this clearly relates to the printing industry. Unfortunately, what they should write is pixel per inch. because dot per inch is different. And pixel per inch is, it means how the size of the pixels in your image, basically. So it's unfortunate, but that's the way it is. So very often when you go to the journal recommendation, they're going to talk about DPI when that should be PPI, okay? So it's okay for you. You will just consider that when they say DPI, the dot is not the printing dots, it is your pixels. So when we talk about 300 dpi, it actually means 300 by 300. It's the same thing as when we say our image had 512 pixels. It's not 512 pixels, it's 512 by 512, okay? So then in that case, we'd have 90,000 pixels per square inch. So nature recommends 300 dpi minimum, and they mean PPI. And Cell and JCB recommend 300 dpi for a color image without text, and 600 for if you have things like a scale bar or text or some arrows or something. So why is that? Why do they make a difference? When we zoom into the image, our images are acquired by pixels, by elements, like we're going to one place, all the sampling process. So this is the reason why when we zoom into our images, we see little squares. It's because each square corresponds to an XY position and an intensity. But all the annotations that we have, they are not images that were acquired with a camera, and therefore they should not. When you zoom into the image, it should be smooth. You shouldn't be able to see squares in there, and this is why there is a difference between all the annotations and the image themselves. So there is a way, and I think this what is written in the recommendations is left over from the time when we didn't have vector graphics. So we have vector graphic now, and if you save your image as a vector graphic, it means that the annotations are actually on top, as a layer on top of your image. And when you zoom in, this is not going to look pixelated. So most of the time, if you save your images, if you have images at 300 dpi, regardless of whether you have a scale bar, arrow, and text or not, you're going to be fine, okay? As long as you save it in vector graphic. I will come back to it. So the size of the image in a figure is going to be printed that way. So it's going to be the physical size in the article. When you get the journal, the paper in your hand, and you measure it, it's going to be that size. So always check the journal requirement. They give you the width of the column in their paper, and that may be different from one article to another. So if we look a little bit, if we have an image that was acquired at 512 by 512, and you want it to be at 300 dpi, which should be ppi, then it means that we have the image physically measures, when it's printed, it will physically measure 512 divided by 300, 1.7, and this is 1.7 times 1.7, and it's in inches, so it is 4.3 by 4.3 centimeter. Okay, so we have an image, and it's going to be inside a figure, which will be in one column, right? So when we want to look, if we want to look at the size of the image in Fiji, we can go to Control I, and we see here 512 by 512. We can also see this at the top of the image as well. So if you can see that if you have if the image has the scale bar and it's not in vector graphic, then 600 dpi are recommended, and then in that case, this image will become 2.17 centimeter by 2.17. It will be very small. Okay, so if you want to publish something in an article, try to get some images that are more than 512 if possible, and also always use vector graphic, because you should always have a scale bar. So sometimes you have a scale bar only in one of the images of the figure. So you should save the stuff in vector graphic, and this way you're good, and you use 300 dpi, and then you're good. Okay, so if you try to acquire the images for publication with a higher number of pixels, if you can. So you have leeway afterwards to play with the size. So when we make figures in Adobe Illustrator, Photoshop, Inkscape is free, so you can use that, or Omero figures. This is where you should make your figures, not in PowerPoint. PowerPoint is not meant for that, and PowerPoint is meant for presentation on the screen. And for that reason, what they want is they want it to go fast. They don't want the images to slow down the presentation, and so they compress everything. Okay, so do not use PowerPoint to make your presentations. Use Illustrator or Photoshop or Inkscape or Omero. There's a very nice video showing you how to use Inkscape, so you can use that, and this is what it's meant for. So if you resize things or you rotate things in PowerPoint, the images are modified and the format compresses the images. So I don't know if any of you, certainly I have made in the past, a long time ago, some posters in PowerPoint, and then when I print it, it looks like, I'm not telling you what. Well, this is the reason. So do not use PowerPoint for that. So only use PowerPoint for presentations on screens and not for to prepare things that will be printed. When you save, this is in Adobe Illustrator, you have different options to save. And the standard is not as good as the high quality printing. So please save with in PDF with high quality printing. And PDF is a vector graphic format. So then you don't have the problem of the pixel, which I will talk about now. So in the printing world, there are two types of images. There's the raster or bitmap images, and there's the vector images. The raster or bitmap images are those that we acquire on the microscope. So they are made of pixels. And the vector images, they can be scaled up and down without pixelation. So you can see this is good for text, for graphs, for arrows, scale bars, and everything. So you can see here, if we have a rastered image, if I zoom in, I will see all the pixels like this. But if I zoom into this image, I will not see. So this is a vector graphic image. That's what we want. And if you use vector graphic, then you can you don't have to use 600 dpi. You can use or ppi. You can use 300. So we can combine the two types of layers. For example, if you put a scale bar on top of a microscopy image, you have the microscopy image, which is pixelated. It's a rastered image. And then you have on top of that the scale bar, which is vector graphic. And then you can flatten them together. Sometimes it's called merged, embed, or it's called burnt. It depends on the software that you're using. And then you will not be able to separate them again. And when they are like this, then when you zoom in, you start seeing everything that was vector as a pixel. Or you can save them as separate layers. So the microscopy image is a raster image. They will only be accepted by the publisher if you have the vector layer on top and they are not flattened. So do not flatten your images when you save them. Okay. So all the graphs and everything must be as vector images, and the flattening, you choose this when you save, and it depends on the format. So if you go to the software that I recommended before, like Inkscape and Omero, you won't have that problem. You won't even see the problem. So Omero Figure exports PDF files as raster with the overlay saved separately. So when you saved your PDF this morning, it was already saved in the correct format. You can directly send this to the publisher. And later, people can work. So the publisher can work on the layer on the vector graphic image. So you can also modify it. So it's very useful to have this format. So do not save as TIFF in Omero Figure because that is flattened. So only save as PDF. So this is what happens when you zoom into the raster image. You can see that the scale bar is not pixelated, but the image is pixelated, which it should be because this is the way we acquire them. Another tool that is very useful in Fiji is the biovoxel figure tool. So you can use that as well. And so you can add the scale bar because Fiji reads the metadata. So if your images contain the metadata, you can add the scale bar without problem. You can put annotations and everything. And there with this biovoxel figure tool, which is a plugin, so you have to download it as a plugin, but I think we have asked you to do this at the start of the course, so you should all have it now. So then you can save the images with the annotations as a vector image, so not flattened. And then you can build in Inkscape, or you can use a mirror figure. So yeah, most of the time when you resize an image, it will lead to extrapolation of data. So do not like resize and then say, oh, I want it a bit bigger, and you resize again, right? So what you do is that if you want to change the image again, you go back to the original and you change to the size that you really want. So in Homero figure, this is the great advantage because you're working directly on the original images. You can zoom in and out of the image. It will only work on the original image. So that is a great advantage. This is all seamless with a mirror figure. You're doing this in the background. So when you resize the image, we can change either the number of pixels or we can change the size of the image if you do this in Fiji. And that will change the printing resolution, which is called DPI, right? Unfortunately. So when you resize an image and you change the number of pixels, clearly the image is going to be smaller because the pixel size is not changed. You don't want to change the pixel size. So these three talk to each other, let's say, so one is deducted from the other three, so you can change two of them. And to reduce the number of pixels, you can go in Fiji to image adjust size, but the quality will be worse. So in general, that's not what you want to do. It's better to do it in Homero, for example, if you can. And you do not want to increase because, you're just filling up the data. It doesn't change anything. So the only way to get a larger image with the same DPI is to go back to the microscope and reimage. So this is something you want to really think while you're acquiring. Get some images that are more than 512 by 512 so that you can publish them. Those that you want to publish, not for the data. So this is a very nice YouTube video by Jan Brochet about the resolution and the scientific figure creation, where he explains all the stuff about rotation and all that thing. So it's very nice. So I recommend this this video. It's not very long. When we resize images, we have the same issue that we have when we acquire images. Sometimes we get moire, and you can see these moire effects in here. Due to changing the resolution in the image, because now if we downsize, then suddenly we're not Nyquist anymore. And so we start seeing some moire artifacts, but this is only in this type of sample where we have a structure. So there's a very nice checklist published in 2023 that you can find. It's called Community Developed Checklist for Publishing Images and Image Analysis. So it's not only for images, but also for the analysis pipeline. What is it that you should think about when you are publishing, when you're writing your paper? And there are like 3 levels, like minimal, recommended, and ideal. And so this is really nice. It's presented like a checklist, and each of the points is explained in the article. So you can use this checklist when you want to publish and make your figures, to make sure that you don't forget anything. Let's keep track of all the processing steps for each image. You can imagine this is a big work. It's a lot more work if you have to find this out afterwards, a month later. So set up a system straight away for how to do this. And I really like this quote from Douglas, who will give a lecture today about ethics. He says, users can do just about anything to a digital image as long as they provide a detailed protocol for what was done and retain the unaltered data, the raw data. If the protocol is out there, they might be called an idiot based on their image processing protocol, but they cannot be called a cheat. And you might think that this will never happen to you, and I am quite sure that it will happen to some of you because this is not uncommon at all, and it's very, very difficult to have your paper scrutinized by someone who asks questions about the integrity of your data. Sometimes, and quite often, it's many years after you have acquired your images. So make sure that you safe-proof yourself against this sort of things. If this happens, it can happen to anyone. But at least you should have the answer. You should have, no, I've done that and that and that, and might be wrong, but as long as it's published, it's fine. And here are the original images. You do not want your figure certainly to be rejected because the images you acquired 1 year earlier are overexposed. So these things are important. It can really be very, very tough when this happens and you're under pressure to publish. Okay, so that's, I think, what I wanted to do, to say. I'm just looking at my timing. Oh, I'm pretty good. So now I want to ask you guys if you have any comment. This lecture is, there's lots of different bits in it, but what I want is to give you some sort of like some tools. You can go back to the lecture to look. Now you have a number of, you can go to the checklist and things like this. So I think that hopefully will help you when you want to build your figures. Maria, you had something?
Yeah, I didn't understand when we need the image rasterized and when we need it vectorized.
So basically, the images that you acquire, they are raster images, always, right? Because they are pixel-based. We are measuring the intensity at sampling points. So the images that we acquire in light microscopy are always raster images. Except, and there's always an exception, except if we acquire storm images, for example. So we don't need to go into the details, but storm images are actually vector images. So apart from that, all the images are rasterized. What you want is that when you're adding the scale bar, the annotation and everything, when you save the images, you want them saved as a PDF. Which doesn't make a raster image. So you do not want the two layers, so the layer of your image and the layer of your annotations, to be burned together. You want them to be saved in two different layers. And that happens when you save as a PDF file.
Okay, so with the PDF, I will save my data as it is rasterized and the labels vectorized.
Yeah, what happens is when you save as a PDF, it saves as a vector graphic. So the whole file is being vector graphic, but the pixel information in the raster image is preserved. So it means that even as you zoom in and out, you will still see the squares of the pixels.
Mm-hmm.
For the annotations, you will not see that. So the image is anyway vector at the end. But it will consider your image as a raster image. So it's a little bit, yeah. I don't know if it's more like a semantic type of thing or not. But when you're saving as a PDF, it's a vector file. It's a vector image.
But
that's what you want to do.
When I published, I was asked to submit the images vectorized. So I guess this is the
Show the images, not the figures.
The figures, sorry.
You were asked to send them as vector images.
Yes.
Yes, exactly, as vector images, not at raster images. Yeah. So exactly. So if you send them as a PDF, it's going to be a vector image.
Okay.
All the journals nowadays want to have vector images because then they can change the size of the image without affecting the the annotations. The annotations do not look pixelated.
Okay.
They are safe basically when they change the size a little bit to adjust things, whatever it is.
Okay,
So
and this happens automatically if you save in PDF.
PDF, yes.
Okay, thank you so much.
If they want to have the original images, then you send them the original images in the original format.
Mm-hmm. Okay.
And normally, they rarely ask for the original images, but you should save the original images, upload them to a data repository for people to be able to access them.
Okay. Thanks.
So,
Yeah, I mean, this is a question for you, but I guess for everyone else as well. Do you have any like recommendations for good data management processes, like, but very practically in the sense of, like when I come to the microscope, like where do I keep track of all of this information before I save it to a folder or even upload it to Omero, right? Do you have any recommendations that work well for you, I guess?
I mean, uh, sort of, they are more and more. So I wouldn't say that I'm the best person to do that. I stopped research a long time ago, and I also stopped research at the time. And things have evolved a lot since I stopped research. So, but there are many. There's a great concern now, and it's been like this for a number of years now because of the low reproducibility of science. So there's a lot more emphasis than before on data management. Very often, when you ask for a grant, at least a big grant, all the European grants and everything, you have to talk about your data management system. And how are you going to store the data? How are you going to share the data? And how are you going to keep track of all the data around? So how you prepare a sample, how you prepare your stuff. And so in general, when you ask for a grant, you have to justify these sort of things. I think that there's a lot of things, not only online, but videos of talk at conferences where you can hear about data management. There's also lots of things about FAIR data. So I don't know if you have heard of FAIR data. So I have to remember what the FAIR
Findable,
principles. Exactly.
findable,
It's
and
findable and
reproducible,
accessible,
accessible.
findable,
Yes.
accessible,
Mm.
interoperable, and reusable. So there's a very strong tendency for that. This is what I'm saying, that you have to put your data on a data repository so that other people can access them and reuse them if possible. And so if you search, I should have, maybe I can try to find a very good video about data management, and I will put it in somewhere. I will send it to you. I will try to find something, a recommendation. But there's a lot of things online. I don't think I'm the best person to recommend. I think in general, I would say that it is important not to save lots of metadata in the name of the file. It's also very important to do things like draw conclusions on a daily basis. You go to the microscope, you do your experiment, you can have some sort of a small conclusion about that day that you put in your electronic lab book, and you link the data. You find a way. Maybe you have like a small file where you have the link to your day. So do not rely on the Windows dates. They are screwed up very, very often. So don't rely on that. But don't put too much in the metadata, in the name of the file. So I will try to find something for you, or a recommendation for you. I'm just wondering if maybe Augustine has a better thing than me to say. So, Augustine, are you around? No, maybe not. Let's see if he pops up.
Sorry, I just lost you while swapping.
Hi, sorry. Do you have any recommendation, website or anything about data management that you know of? Because I think everybody comes up with their own solution, but they are nowadays quite a lot of streamlined things. Or maybe this is what we would want to recommend instead of right.
Yes,
I have my own system, but it's not very good to look at individual systems. I think it's best to go for the community recommendations.
there are several web pages. But it also depends on which step of the data management. And also, many of the institutes have a data management office, and there's a lot of support on that side. So I would definitely advise, I know Karolinska has the data management office, which you should definitely contact.
Yeah, that's a good idea. I think I will also contact them.
And they have a very nice webpage describing many of the different steps and how to handle all these.
Okay. Okay, but that's a great idea. I may ask them even to give a lecture next year.
Yeah, I can give you the name of the contact person who is often giving lectures on data
Right.
management. Also, well, NBIS also probably has experts that can give lectures on this side.
Yeah.
I'll look for a couple web pages.
Okay, thank you. Thank you. So I will try to find something and Bruno and post it.
Thanks, super helpful. And can I just have like one more quick question,
Yeah.
the follow-up? So of course, I work a lot with like super resolution images, SMLM, and mean flux. And what is like the rawest image that you would save in that case? Because like a lot of these softwares that are, like Zen has a inbuilt, for example, drift correction, and I usually use that. And now I'm realizing that I'm not even saving the rawest data, right? Because I just saved the drift corrected data, and I guess that's wrong, right?
Well, I mean, it's an amazing question. And it's one that has been hurting for many years, I think, lots of people. So it's very difficult to know because one thing that is important is that if you save the drift corrected images, when you publish, you definitely must publish the exact version of the software that you were using when you did that. At the end of the day, I'm not sure who is going to go back to the original non-drift corrected data ever, but there's a lot of things. So now we're just talking about drift correction. How is it made? Was that good or not? To start with, when you go to the microscope and then you push the button that says, I have this fluorophore, this fluorophore, this fluorophore. Sometimes the names are not even correct. And then you put, please set up my whole microscope, and then you don't check even how the stuff is set up. That's also questionable. Now, if what is the original data is actually quite an interesting question, because some cameras have a correction inbuilt in the camera. And nowadays, even detectors have that. So I was saying that nowadays we cannot really change the offset because it's sort of it's calculated. So that is, all these type of things are a layer of correction that the software is doing, and also that can change in the next version. So that's an issue. What is the original metadata? But yes, I think that Agustin put some nice links in there that you can have a look at as well. I think that, for example, if you take an image that is light sheet data, and sometimes in some light sheet systems you have to rotate the sample and take another data set. Then afterwards you fuse them together. These data sets are very big. If you take 3 rotations and then you fuse them, you have 4 times the whole volume. It's very big. But and you have to save them for 10 years. So it's a little bit difficult for me to give you an answer, because I think that the whole imaging community is actually wondering about that as we speak. And I don't think that there is a very clear answer for that. I would say that maybe in your case, you could save the data before the correction. You can always apply the correction afterwards. The question is, can you go back? And this is something you can ask the company who sells your microscope. Can you go from the corrected image to the previous data? If you can do that, then it's fine to save the images that have been drift corrected.
Yeah. Yeah, a couple of very good points. Thank you so much. Super helpful.
Ken, you had a question.
Yeah, I do have a question because nowadays I'm working on the publication. It's like if I want to assemble the images into a figure to fit the size of the panel, I have to resize the images, like to make it larger or smaller. Do you have any suggestions when I resize it just in the Adobe Illustrator or in
I think you should go to our wonderful Homero figure.
Yeah, Omeros. Yeah, but
And all the
the
problems
size
disappear.
matter.
Yeah. If you do this, if you have to if you have to resize several times, do not resize several times with the same images. Yeah, go back to the original, do it again. If you do all this in a mirror figure, then it's fine to resize several times because they are working on the original data. You only leave the original data when you press the save button in a mirror figure. So this is the beauty of this stuff, is that you're actually working on the original data when you make your figure. So I think this is a bit of, it's a very nice solution because it's a bit fail-safe. If you work in other software, you need to have quite a lot of knowledge, let's say, to avoid these pitfalls. And again, it's the same thing. If you have very big things that are very obvious in your images, you rotate them or you change the size or something and you create all sorts of weird artifacts. Nobody will see it. But if you have small details, then it's important.
Okay,
Yeah.
thank you.
You're welcome. I hope that you get your paper published, Jen. Sophie.
I also have a question because you scared me with the whole someone will come to you in 5 years and scrutinize your paper. But I was wondering if it's normal or if it's happening that you publish a paper including some imaging data, and then you take a course like this and you realize that you did something wrong. Do people then go back and correct it, or
So correct it, what do you mean? Reacquire images, or what do you mean?
I guess if you haven't done something completely as you would after this course,
You mean
and
you
you
have
think,
published some data?
and it's already published,
It's
exactly.
already published, and now you go like, ooh. Yeah.
Yep.
I think that it depends on the severity of the thing. If you think that the interpretation of the data is really off, it wouldn't be a bad idea to actually to try and correct things, I think, if you think that the interpretation is completely wrong.
And is that something that people actually do?
Well,
I haven't published anything. I'm asking in theory.
I think, is that something that people do? I think as we develop as researchers and everything, we learn things slowly, right? And it's normal that the things that we do at the beginning of our career is sort of slightly less polished than the thing that we do at the end. What you do not want is that someone comes up to you and says, "Hey, look at these images." And if they just say they're not nice, is one thing. If they say you didn't acquire them in the right way, but you have published the way you acquired them, it's one thing. But if you do not write how you acquire them and the conclusions are wrong, and someone comes up after you, that is not fun. And that can happen many, many years afterwards. So that's what you want to try to do, is to create a fail-safe such a way that you publish your images. There's clearly in science a lot of things that are published that are not correct, because this is the way science moves forward, right? So, but it is not rare. It's not rare. People tend not to publicize when they are being sort of pointed to, and when they get in trouble that way, they don't tell everybody, but it's actually quite common. So it is not fun at all to be scrutinized in that way and to have to try to dig into something that you published years before. What you want is, in that case, if this happens, that everything is in place, that you have a good system. So by any means, think about it now. How are you going to make sure that if you have something that is published and in 5 years something comes and asks you a question, how are you going to figure out how this was done? Right? So set up a system that works, and I will find some links that are nice, and I will share them with you. Yes, we had Bruno maybe who's first.
Sorry, and let me know if I'm taking this discussion way too far. But I'm just wondering, even if you report everything that you did, like up to the nitty-gritty stuff, I mean, even reporting like 3% laser power for this fluorophore. I mean, that still means like 3% laser power of your laser, of your microscope that's like maybe 10 years old. And those are like things that you're not reporting.
Yeah.
And I'm just
So,
like going in a spiral of like things that I cannot control. And I'm almost certain that if I brought the same sample to the same microscope in a different facility, I would not get the same image.
Absolutely.
If I have,
Even to the same facility.
if I have an opinion, all of this is just reduced as much because you cannot replicate it exactly as the same.
The idea is not that you report everything. Exactly. As Augustine says, the lasers grow old and everything. The percentage of the laser is in the data, in the metadata, right? So you do not have to report that. You just must have the information ready if someone asks you. So you have the image, the information, the image saved in the correct format. This information is in the metadata. As you said, knowing that it's 3% of the power will not help this person reproduce the data. So you should not report this. You should just say that you have set the settings of the microscope to have the best signal to noise ratio, for example, and also to have no saturation, no underexposure. If you set this, it's enough. If someone wants to know exactly how you image, they can ask you, and you have this information in the image. So you don't report this type of things. You only report the things that are important for the reproducibility. So someone else in another lab, maybe 10 years after you, they want to do the same thing, and then you have the data there. It doesn't mean that you write the protocol for them. It means you write the things that are important. I use this objective, it had this numerical aperture, and it has this immersion medium, right? And I was using this type of microscope, and I was using the spectral detector, and I had this filters. You don't even need to say that you had a spectral detector or some physical filters. You just set the filters. Then if they want to use a spectral detector, so now that you have knowledge about the machines and everything, I think you're able yourself to judge what you would need to know to reproduce the data or not. And that's what you put in your paper, and you make sure that everything else is nicely organized in your folders so you can find it. Christos.
Hey, I had a question about the visualization of the images in the context of a scientific article. Correct me if I'm wrong, but in the big majority of the articles I've gone through, the images, even in the single channels, they're colorized, like they have colors anyway. And is it something that that happened by accident? Was it in the past different guidelines, or like
I think in the
what?
past there were no guidelines.
Okay.
In the past, maybe 10 years, I would say, even more so in the past 5 years, there's a very big challenge where so there's a lot more, so there is an issue with reproducibility in science, and that is clearly documented and published. For example, in I don't remember the number, but I think one paper that I read where they were looking at images of microscopy in, I don't remember, thousands of articles. Only 20% of the articles actually had in the material method enough information for people to reproduce the data. So there's an enormous problem there. And I think that the community is more and more aware of that. Journals are more and more aware of that. So they're more demanding. So I think it is very important not to look backwards, because this is where things were not well done, and to look forward, because definitely it's going to become more stringent. So you have to make sure that from now on you are setting your things correctly, because it will be more stringent in the future.
Okay. Yeah, that's good.
And it's very, very unfortunate that we have all, because for us, when someone comes to us and say we want to use this microscope, I would like to reproduce this article, we look at the material method and it's appalling, the type of things that you see.
Yep.
So you're going
Exactly.
to have to write your material methods.
Okay, great.
It's one assignment.
I was just thinking for a second that if we submit an article with images in black and white, maybe we will look like the exception. And yeah.
Yeah, this is, so did you see the paper, the recommendation for how to publish microscopy images and also publish microscopy pipeline is only 2 years old.
Yeah. Okay. Great, thanks.
Good. I wonder if there's anything else. It's nice to have this conversation, because I think that these things are not discussed enough.
Yeah, it's not. I have only one thought of mine, because just two days ago, an article in which I did figures was published, and now I see the figures. It's at least the question can still be, even though I've learned a lot of ways to improve the images, I feel comfortable that at least with the kind of analysis I do, it did not impact a lot. So I guess the results still stand. I'm not feeling worried about that, but now I feel even more happy and confident that I can improve all of this that has been done. Yeah.
Yeah, exactly. So as I said, it's unfortunate that the science works the way it is, that we have to learn so many different techniques by ourselves. And so everybody makes mistakes, and this is why we are here and we have this course. And it's the same thing with all the other techniques. You wish you could be better trained, and then you would know what you were doing. But this is the way, unfortunately, things are, and maybe it will change in the future. I'm not sure. We have to accept the way it is. What is important in all this is that if you have published images that were wrongly acquired or whatever, or wrongly shown in the past, as long as you have all the information with you and you know where it is and you have the original images, you're safe. You don't have to republish all the papers that you have. Just think about it in the future and do the stuff in the correct way, and slowly we will manage to get the ball rolling and change the world. Oh yeah. Okay, are there more questions? Okay, if there are no more questions, we're going to go to the the workshop Zoom, and we're going to write our material methods. You guys there.